Thank you, Matthew. Good afternoon, ladies and gentlemen. I'm going to present you over the next few minutes Promethera, technology, business model, and the clinical results, midterm analysis of the 6 months follow-up of the ongoing phase 1/2 of clinical trial. So let's start by the unmet need. As Matthew said, we are a liver-focused company, and you see here an example of a disease we are targeting currently in clinical trial. You see on the left a premature newborn that has jaundice and is under UV. Is under UV because the liver is immature and has not the ability to conjugate a specific pigment, a yellow pigment called bilirubin, that provides this yellowish color because it sticks to the skin. And the UV can destroy the bilirubin from the skin and avoid the brain toxicity because this pigment, bilirubin, is highly toxic for the brain. Fortunately, in the majority of cases, the liver becomes mature after a few weeks and you can relapse the treatment. In some cases, the enzyme in the hepatocyte that conjugates the bilirubin, which is excreted in the bile, is mutated. So the kids will never be able to conjugate bilirubin, and it's what you see on the right. In this case, it's a little girl. She needs to spend 10 to 12 hours per day under UV to avoid brain damages. And if she does not, if she go in holidays and forget the lamp, she can suddenly die from brain icterius. So they are really in jail. So the only way to cure this kind of patients today is liver transplantation. So you need to change the full piece, as just one little enzyme in the hepatocyte is missing. So what we want to do with Promethera is, as you know, there is shortage of livers, is instead of changing the liver, is to infuse stem cell, allogeneic stem cell coming from the liver of cadaveric donor in the liver of this patient that will regenerate the enzyme. So we do enzyme replacement, as John Shire is doing, but using the vector, the cell as a vector instead of infusing recombinant proteins. So there is more than 300 different genetic diseases that can affect the hepatocyte, the enzyme of the hepatocyte. Each of them are very rare, ultra-orphan, and we have selected 2 of them currently in clinical trials to do the proof of concept of the platform, because the cells can be considered as a platform. Let me explain you how it works. No, it's here. So you see here the platform we have. We have a GMP facility in Mont-Saint-Guibert in Belgium, in the south of Brussels, and we can, from the hepatocyte bag, which are coming from the liver of cadaveric donor. There's a tissue bank that has been founded by the scientific founder of the company, Professor Etienne Sokal, in Clinique Saint-Luc. They can collect these livers and process them in a very easy process in human hepatocyte bag. And this human hepatocyte bag are the raw materials we are using in our GMP facility to purify and scale up in very large quantity the stem cell present in very small quantity in the liver. And thanks to the scale-up capability of the stem cells, we can, with one liver today, we have the ability to cure more than 500 kids instead of one liver, one kids with liver transplantations. We have been quite innovative in terms of manufacturing. I do not present anymore the GMP van that we communicated in the past because it's obsolete. We are now a second generation for the formulation. So at the end of the manufacturing process, we are freezing the cells in a cryopreserved in a specific vial, and these vials are resistant to cryopreservations. And now this system is completely validated. You can, the doctors, the pharmacists of the hospital can reconstitute himself by just taking out this little vial from the fridge in 2 hours or even 30 minutes, the cells with the diluent and infuse through the portal vein the cells that has been freshly reformulated. What is important to know, as I said, it's the cell can be considered as a platform because one healthy cells coming from a healthy donor have all the enzyme functioning. So with exactly the same cells, we can treat, in theory, any of the 300 different genetic diseases. Of course, we cannot do 300 clinical trials, so we have selected 2 of them, and the strategy of the company is to do extension of indications after market approval for 2 of them. Just a few words about the company. So we are a phase 1/2 clinical trial, and I will come back to that. We get approval for the next pivotal clinical trial, phase 2B/3, and in Q4, in a few weeks ago, in fact, in Belgium. We have been founded in 2009, a team of 48 persons. We raised close to 40 million euros over the last 2 rounds of financing, and we will raise another big one, I hope in 2 weeks, even last week's, but I don't want to communicate today. And we have been able to attract very interesting investors with Vesalius Biocapital, the Japanese Mitsui Global Investment, Shire, Boehringer Ingelheim Venture Fund, ATMA Pile that do the bioreactor that we are using for the scale-up of the manufacturing. So the pipeline. So today we have selected 2 indications, which are Crigler-Najjar and Urea Cycle Disease. Urea Cycle Disease is a specific disease where one of the 6 enzymes of the urea in the hepatocyte is mutated. And in this case, when the kids eat proteins, there is accumulation of ammonium in the blood. And this ammonium is, again, highly toxic for the brain and use brain damages and finally death for the kids that are not properly treated. So we have selected both indications. We have completed, and we will complete the 12-month follow-up, in fact, this year. For the phase 1-2, and starting for the urea cycle, the phase 2b pivotal in pediatric indications, and both are in pediatric indications. But we have, as an overall range of indications, we would like to target maybe next year, like hemophilia A or other indications related to inborn error of metabolism. We have seen that our cell can produce factor 8, factor 9, factor 10, so it opened the door of the hemophilia market. And something we could develop later, alone or with a partner. And we have as well another range of disease, more the adult disease, with a different mechanism of actions. Here it's not enzyme replacement that we are targeting, but inhibitions of liver fibrosis and replenish of the stem cell pool that has been destroyed by the toxic in liver fibrosis or acute and chronic liver failure. So the team, so we have a quite good management team. So we have half of them coming from GSK Vaccine in Belgium. The headquarters is not very far away from our facility, so it's a cluster effect, so it helps to attract very talented people. And it's the case of the Chief Medical Officer, of the Chief Operating Officer, or the Regulatory Affairs. Etienne Sokal, the scientific founder, is as well a key opinion leader in pediatric hepatology. And we have recently a CFO joined for the future development of the company. So business model, we are like, once again, Shire, Genzyme, Biomarine in the ultra-orphan, meaning very small number of patients. So you see for urea cycle disease, we have 3,000 patients in Europe, 3,000 in US, maybe 700 in Japan. And for Crigler-Najjar, it's even less, the number of patients. But if you add all the 300 different genetic defects, it becomes a very big market. So once again, today we select 2 of them just to do the proof of concept, and later we will expand the indications with the other one. In this market, the ultra-orphan pricing, it's still today in US and Europe market where you can get price premium. Of course, with good data, good clinical benefit demonstrated. And it's the case you see here, a benchmark of different enzyme replacement therapy, which are for the majority of them are biologics, and you see the average yearly price or yearly cost is around 100,000 per year. The cost of a liver transplantation in US, which is today the only way to cure some of these patients, is $500,000. So we are really in disease where the unmet need is huge, life-threatening, pediatric, and where the alternatives are extremely costly. Okay, let's go now to the clinical trial. So today we are conducting 2 clinical trials in parallel. An investigational study where we are infusing the cell, keeping the patients with the standard of care, which is UV for the Crigler-Najjar, or low protein diet and scavenger of ammonium for UCD. And we have in parallel a control group called observational study where we follow patients with just the standard of care only to reinforce the validity of the difference of evolution of the disease between the cells, the patient with the cells and the patient without the cells. So it's a dose range, dose escalation study where we follow the efficacy, but the safety and preliminary efficacy on 20 patients. So we have enrolled 14 UCD patients and 6 Crigler-Najjar. And as you can see, we infuse, all of them have been infused through the portal vein with low, mid, and high dose. And you see here a snapshot of the result. So I can start to show them because it has been already presented in 2 international scientific conferences. So what we wanted to do in this specific clinical trial is first demonstrate cell engraftment in the liver. And it's feasible in the liver, in liver disease, because you can do biopsy. And with this biopsy of the liver, prior infusion of the cells, 6 months and 12 months post-infusion of the cells, you can look for donor cells present in the liver, and that has been done with different techniques, digital PCR, but and immunochemistry, and we have clearly demonstrated the presence of the cell 6 months and 12 months post-infusions. We wanted to show the long-term in vivo potency of the cells, so the ability of the cells to conjugate bilirubin and to produce urea, de novo urea, which is missing. And here we have been using a specific isotope of the sodium acetate, which is the substrate of urea cycle, I will come back to that, to demonstrate the apparitions of urea C13, which is the de novo productions by the liver, thanks to the cells you have infused of urea. So it's really a clear readout and demonstration that the cells that you have infused are functional, and you will see the increase we have seen 6 months post-infusion of the cells. The last element are the clinical benefits, because at the end of the day, you need to show the clinical benefits that the cells produce to your patients. And here we have seen a statistically significant decrease in UCD patients in terms of ammonium. We have seen as well in Crigler-Najjar patients a decrease between 40% and 10% in the terms of level of unconjugated bilirubin. So quickly, because I just have 2 minutes from what I've seen, I will go to show you some pictures. So you see the biopsy here before infusion of the cells, so you have nothing. So when you have null mutations, you have really no protein expression at all. And you see 6 months post-infusion of the cells, the spot with the proteins through immunochemistry present in both patients at 6 months. We have as well used the urea C13 test. So the urea C13 is given per os to the kids. And when the liver and in the urea cycle is functioning in the hepatocyte, you have the C13, which is a stable isotope of the C12, which is incorporated in urea that you can follow through mass spec in the blood. And in normal volunteer, in adult, you have this kind of kinetics. So if you take blood sample every 30 minutes, there is accumulations of urea C13 that reach a plateau and decrease slowly. And you can measure the area under the curve. And you see here an example of one of the patients of the study we have infused with the cells. You see before infusion of the cells, the baseline is extremely low because in normal volunteer, you go up to 4. 3 months post-infusion of the cells, 6 months post-infusion of the cells, the area under the curve has been multiplied by 7-fold, so which is a significant increase in terms of capacity of the liver to metabolize urea. In terms of clinical benefit, an example for Crigler-Najjar, you see here before the fluctuations of the unconjugated bilirubin in the blood, which is extremely high, and post-infusion of the cell, you see this decrease. And for this specific patient with the lower mass of cells, we have a decrease up to 1 year of 28%. Okay, just to close, let me show you the clinical development plan. I can? Yeah, thank you. So you see here just for the 2 core indications, Urea Cycle Disease and Crigler-Najjar, we are working on the new indication plan that I presented earlier. So the idea is to start the 2 phase 1/2, phase 2b/3 this year, beginning of next year, and to be able to submit based on interim analysis to the EMA in 2016 to potentially get approval end of 2017. Conditional or real approval will depend of the final data we will get. So on this plan, and we have as well forecasted to do a reinfusion protocol to reinfuse patients 1 year post-infusions. And I would like to thank you for your attention and just a snapshot of the team in Belgium under the sun. Thank you.